about what we believe are the mechanisms, who is most at risk of developing chronic pain, and why we think that might be. I think you probably all know someone who suffers from chronic pain. Back pain, arthritis, migraine. It's a very common condition and affects about 1 in 5 people in the general population worldwide. In fact, Poland apparently is quite unlucky. Your incidence is reported to be at 27%, which is slightly higher than the European average. On the bright side, the Polish population suffer from pain slightly less long than the rest of Europe, 6 years on average compared to 7 years, and a lot less than the poor Finnish, who report suffering from chronic pain for 9 years. So you can imagine that these are really very devastating conditions. And what is particularly sad for us in the clinic is that we cannot really provide many treatment options. And this is despite the fact that we actually understand quite a lot about what causes chronic pain mechanistically. It's believed that in the body of a chronic pain patient, you have hypersensitivity at all levels of the nervous system. So you have the peripheral sensory neuron that carries information from the body to the central nervous system. It responds more than it ordinarily should. Then you have the spinal cord, where there's lots of abnormal inhibitory interneuron circuitry. And finally, you have the brain, where the pain percept is generated, where again, you have abnormal activity. I think someone's trying to get in. Okay. Hi. So, on top of this nervous system dysfunction, we also know that you have abnormal immune system function, both in the periphery and also in the central nervous system. For instance, in the central nervous system, you have involvement of microglia. So these are the phagocytic immune cells of the CNS. And here is a preclinical example. This is a spinal cord of a mouse sliced horizontally. The microglia are stained in turquoise. And I think you can appreciate that there's a lot more microglia on the right side of the spinal cord. And this is because the authors of this paper have injured this mouse. They have cut the nerve all the way down in the thigh of the mouse. They've cut the common peritoneal and tibial nerves. And this injury all the way out here in your body will make the nerves here, which are bipolar, so they go from, have an axon down here and an axon up here. And the axon up here in the spinal cord will release factors, including CSF1, that recruit and activate microglia in the spinal cord. We know that these mice are in a pain-like state because we can measure, to some extent, how they respond to evoked stimuli. For instance, you can use little filaments to poke their paw and measure at what point they withdraw their foot. In a normal mouse, they normally withdraw their foot at about 0.6 to 1 gram. And after injury, you can see this blue line here. They're much, much more sensitive. 0.2 gram is enough to get the mouse to respond. In contrast, the red line is a mouse that has CSF1, this factor that recruits microglia, deleted from just sensory neurons. And you can see that this mouse doesn't mind at all about the poking of the foot. So we've essentially abolished the pain response, the mechanical hypersensitivity, in response to really quite a severe injury, simply by not involving the microglia up here in the spinal cord. So this is something that has been shown time and again by many groups. The effect sizes may be a little smaller than shown here, but it's certainly something that we believe to be true, at least in preclinical models. On top of the mechanisms, we also know quite a lot about who is more at risk from developing chronic pain. Unfortunately, some of us are more likely than others. And even more unfortunately for me, being a woman is a risk factor for chronic pain conditions. And unfortunately for all of us, being older, becoming older, also increases your risk of developing chronic pain. About 57% of the general population in Poland above 65 suffers from one kind of chronic pain condition. Other things, other risk factors, I mean, this list is not exhaustive, include adverse life events, in certain personality traits, like catastrophizing. So in my lab, what we've been trying to do is to somehow link these known risk factors back to mechanism. What is it about these risks that make you more likely to develop pain? How do they tie in with the mechanisms that I have just told you about? On a very generic level, you could of course say, well, there's got to be either genetic or environmental causes, and some of the factors may be grouped quite neatly. Obviously, adverse life events, it's something that happened in your environment that interacted with your body and made you more likely to develop pain. But what is that and how does this interaction work? And quite frequently, we have something in the middle, a gene-environment interaction, where your genetic makeup together with your environmental experiences predispose you towards pain. The genetics of this disease have been studied quite well, so I'm not going to talk about these. What I'm going to talk about is the mechanisms in this space here. How could the environment impact our genes to make us more vulnerable to develop chronic pain? And the mechanism that I'm going to talk about is epigenetics. A lot of you will be familiar with this. Epigenetics are changes to gene function that do not affect the DNA sequence itself. The classical ones are DNA methylation, so your DNA can have methyl groups, and the position of these methyl groups affects how it can be accessed and used. And on top of that, when your DNA is packaged to fit inside the nucleus, it is wrapped around these histone proteins, and they can also be chemically modified. And the nature of these chemical modifications determines how the sequence can be accessed and what transcription factors are being recruited, so how the sequence is being used. What is special about these canonical epigenetic modifications is that they are heritable in dividing cells. So this means that if one cell acquires a change, all its daughters might still have that change. So you can appreciate that this is really quite a long-term mechanism that could confer risk, so to speak, over long periods of time. What is important to remember about epigenetics is that epigenetics is what determines cell type. A neuron is a neuron because it can access neuronal genes. And obviously, your skin cells don't suddenly turn into neurons unless you make them so through stem cell technology. So fundamentally, once development is complete, most of your epigenome is static. And it also means that most of your epigenome, or all of your epigenome, should be studied on a cell type specific level. Right, so I told you your epigenome is static. Why do I care about it? I study chronic pain, a disease mainly in adults. I care about it because lately, over the past 10, 15 years or so, it has become clear that a small subset of your epigenetics can still be impacted even in adult cells. And it seems to be impacted mainly by environmental components. Here, for instance, is an early pioneering study where they looked at identical twins. So identical twins have the same DNA and the same epigenetics because it is thought that the zygote confers the epigenome from the mother. So obviously identical twins share a zygote. And so they find what they expect here. This is global histone acetylation in blood. You have 3-year-old twins have the same acetylation level. But then when you look at 50-year-old identical twins, differences start to emerge. And this has since been repeated with technology, has advanced quite significantly, has been repeated several times. Here we have a couple of studies looking at DNA methylation in identical and non-identical twins, and they show that the variance in DNA methylation, the difference in DNA methylation in these twins, is largely explained by non-shared environmental variance. So it seems that epigenetics could be a conduit, a player that allows the environment to impact gene function over long periods of time. So then in our context, we could hypothesize that all of us start off with cells that have a particular genome and epigenome that in and of itself will already confer some risk, right? You might be lucky with your genetics or unlucky. And then you live your life, and sometimes bad things happen. And some of these bad things might be salient enough to actually leave behind a few changes in your epigenome. And over time, these small changes could accumulate and lead to long-term sensitization of the nervous system of the type that we observe in chronic pain. So that's really the hypothesis. What is the evidence? I have to say at this point that we're really at the very beginning of investigating this. So there's not that much evidence out there yet. But I'm just going to show you one example from my lab that suggests that some of this may be happening. We are interested in enhancers, and we use the epigenome to find where they are. So enhancers are regulatory regions of the genome. So here, for instance, you have a cartoon of the DNA sequence. Imagine this pink region is an enhancer, so it could control gene expression much further away. But this particular enhancer is really tightly packaged, so it cannot be accessed. It's inactive. Other enhancers are what's called primed, meaning that they're accessible to transcription factors. They're not necessarily always bound by transcription factors, but they're open. There is space, right? There's potential for usage. And these primed enhancers can be usually identified by having a single methyl group at lysine residue 4 of histone 3. It's kind of a mouthful, but they have a particular mark, chemical modification of a histone that is close to them. Again, mostly enhancer profiles are set once cell development is complete, but the immunologists have shown us that enhancer remodeling in adult cells can be very important for certain types of immunity. So adaptive immunity in T cells, for instance, is conferred through enhancer remodeling. And a few years ago, it has also been shown that enhancer remodeling can also occur in innate immune cells, namely macrophages. Here's an example from bone marrow-derived macrophages. They're putting interferon gamma, a pro-inflammatory substance, on these macrophages. And 72 hours later, you can see here this little blue peak is a novel primed enhancer that has been measured in this particular region of the genome. And what is particularly interesting is that when you wash away the interferon gamma, 48 hours later, your cells look quiet again. They look like normal bone marrow-derived macrophages. But this particular enhancer remains in place, suggesting, if you allow me to anthropomorphize, that this cell remembered that it has seen the interferon gamma, and it can respond differently the next time around. So we saw the last author of this paper present these data in 2012 and thought, well, that's interesting. What about our favorite innate immune cell, the microglia? Remember, I told you about the microglia and its importance in neuropathic pain. And the microglia is, of course, a direct cousin of the macrophage. So what we wanted to know is whether microglia might also acquire novel enhancers, particularly in contexts of pain. So we performed a conceptually very simple, but technically still relatively challenging experiment where you ligate the nerve of a mouse, as I've shown you before, and you extract the spinal cord microglia. So this is what it looks like. We perform FACS, or flow cytometry. First, you gate your single live cells, and then you take all of the immune cells, all of the cells which are positive for CD45. And then amongst those, you take cells positive for CD11b, a generic myeloid cell marker, and P2IR12, which is a marker of resident microglia. And you can see, so the microglia from an injured animal are in red, microglia from a control animal are in blue, and you can see that the vast majority of immune cells in both of these cases are resident microglia in the spinal cord. And we've seen that time and again. There's no infiltration, really. Other people have also reported on this. And this is contrary to what was originally known in the pain field. People thought, oh, maybe this injury out here will create a breakdown of the blood-brain barrier and you will have infiltration. So I think by now the consensus is that everything remains stable. You only have your CNS immune cells here. We can look at what these microglia express by performing RNA sequencing. And after the injury in the thigh, you can see that not only do they proliferate, but they're very clearly activated. They are recruiting complement component pathways, interferon in gamma and beta genes, lysosomal genes, and of course genes involved with mitosis and protein synthesis because the cells are dividing. What we didn't find, and this is for the pain aficionados among you, in the pain field there's a big story about how microglia release BDNF. We do not find any significant expression of BDNF message in this microglia. And neither does Marco Prinz. Marco Prinz is a professor from Freiburg who is very famous in the CNS microglia field. He's usually introduced as the prince of microglia at neuroimmunology meetings. He performed something very similar. It was facial exotomy. So instead of cutting the nerve in the thigh, you cut the nerve in the face. And then you take the microglia from the CNS, and then you look for BDNF before and after injury, and you see there is absolutely none. So we don't have any BDNF message. And I think this was quite interesting to us and to a lot of other people as well. I want to take a super slight detour at this point and tell you a little bit about cell type specific sequencing data. And I want to remind you that you can access all of these yourself. You don't need to be a bioinformatician, you don't need to generate these data. They're now widely available on the internet, downloadable as Excel, either from the publication or from GEO datasets, which is just a subsection of PubMed. You can learn how to access these data by following this link. It's about as complicated as explaining to someone how to use primer 3 for the molecular biologists among you. And you can find out what is expressed in sensory neurons, cortical neurons, excitatory neuron subtypes, interneuron subtypes, somatostatin interneurons. I know what they express. Human and mouse astrocytes, astrocytes in aging, astrocytes after spinal cord injury, microglia and every other resident myeloid cell type in naive and many pathological states, aging, stroke, LPS injection, oligodendrocytes in the entire lineage, and every single cell type in human blood at minimum 90% purity, courtesy to an epigenomics consortium called Blueprint. And in fact, a lot of these data come from the epigeneticists because they have to work in a cell type specific space, and they need the RNA sequencing to make sense of their other chromatin explorations. They will provide these data for you for free. So if you don't care about epigenetics, the only message that I'd like you to remember from this talk is that you should use these data and really accelerate your own research for free. Now, coming back to the main question, are there novel enhancers in microglia after nerve injury? This is a little snapshot of a genome. Here are your enhancers in a control animal. Down here is a chemokine gene called CCL12. And here, after injury, you can see that there's a lot more binding at this particular spot, suggesting that there were indeed novel enhancers in some of the microglia in the population that we extracted. We found a small but significant number of these. And what was interesting is that a lot of them were very close to genes that were transcriptionally dysregulated. CCL12 was upregulated in these microglia, and this enhancer is very close to CCL12. So this suggests that there might be some functional relevance. We also found that this altered binding lasted for quite long periods of time. Here we looked 28 days after nerve injury. We still had increased binding at these putative enhancers. And this was at a time when transcription is back to normal at these genes. So they're no longer upregulated, but you still have this increased binding. So at this point, I would say that yes, the microglia mirror the macrophages in a dish. We think that there might be altered novel enhancers in a small subset, and that these outlast some of the transcriptional changes we see. So going back to our hypothesis, we have some early tentative evidence that neuropathic injury can impact the epigenome in microglia. But I really think we need to do a lot more work. A lot of it is being delayed by the technical problems associated with this. For chip sequencing, when they published protocols for low cell number chip sequencing a few years ago, they talk about 100,000 cells. I have 10,000 cells. And now we can do some of this with 10,000 cells, We have done some enhancer function in sensory neurons, but we're trying to push it further, and we're trying to look at enhancer function in hundreds and thousands of cells rather than 100,000 of cells. I'd like to thank my wonderful mentor, Stephen McMahon, the people involved in the work, my funders, and you for listening.
Thank you very much for a very exciting lecture. And if you have questions, and I see one, Professor Paczewski.
Yes, so I mean the injury to the neuron comes first, but then it is believed that the activity of the microglia can be damaging, not necessarily to the original neuron that has been injured, but to all the other neurons that are in the CNS environment. Yes, so obviously they have a very positive function, but like anything, so I think it's going to be a question of getting it right. So like inflammation usually has a protective function. It should be good, but too much inflammation, like in autoimmune disorders, can lead to something bad. And so it's believed that in pain you have something similar. So at first it may be good, but then it hangs around and it doesn't do anything. And actually, with the microglia, it's even more interesting. I mean, obviously what the peripheral neuron is trying to do is to recruit immune cells to the injury site, out here in your leg. It's quite possible that the release of CSF1 from the other axon up here in your spinal cord is an epiphenomenon, something that just happens. It's just leaking CSF1 on both ends, yeah? And maybe the CSF1 that it's leaking in the spinal cord is actually bad.
But do you have evidence that that's what is happening?
Well, we have evidence that from many, many different labs, that if you interrupt the microglia response, either pharmacologically or genetically, you can abolish pain behavior. That is what we have. Preclinically. Clinically, it's a bit more difficult. They're trying to image microglia with something called TSPO. It's sort of, so we're still kind of wondering how much of this is relevant necessarily in humans. But in animal models, this is a big theme right now because you can really impact the pain sensation of the animal in a chronic pain model.
You show in one of the figures that the injured mice or injured rats, then they come back to the values of a normal and healthy animal. So this is what happens when you, for example, have an animal in an experiment for 2 weeks or even 3 weeks, and then so they experience pain. You record this pain, and then the pain is gone once you stop the microglia. That's what you said basically. Locally in retina, right?
When the cells, damaged cells are cleared, everything goes by a state. So they are playing positive role, and I don't see evidence that there's a prolonged activation of those cells.
Well, in the pain field, there is evidence for that, though. I mean, I have to say, I'm not really particularly wedded to the idea of microglia. I chose microglia because I could get them out in sufficient numbers to look at my experiments, and because microglia are the cousins of macrophages. I still fundamentally believe that for really long term, if we're talking like 6 months to a year, of course, fundamentally hypersensitivity has to be lodged in the neurons somewhere. So that's why my diagram. But I think immune cells have their role to play.
Thank you. Do we have more questions? If not, thank you very much again, Fran. And now I would like to introduce our second lecturer of this session, Professor Jacek Kuźnicki. Many of you definitely know Professor Kuźnicki. He is the to integrate biology and engineering in what she's doing in Martin's lab. So please.
Thank you. Hello, everyone. I would want to show you a few examples of data analysis on behavioral studies and also electrochemical studies of neurotransmitters. So machine learning techniques allow us to let computers learn without being explicitly programmed point by point what they should do. And they are, of course, used in robots, in handwriting recognition on your tablets, as well as data mining. And this is what we will be talking today. And machine learning algorithms that are used in data analysis can be basically divided in unsupervised methods, in which we just put all of our data in the algorithm and we don't say anything about it. So then we let the algorithm to show us outliers or to reduce the dimension of our data in order to be easier to analyze it, or to search for patterns. So we can search for some clusters or groupings of the data. In supervised methods, we already say that this sample was coming from a sick patient or a healthy patient, or maybe we measured different concentration of some compound. And then we build a model based on those set of samples, and we try to use it to predict the thing that we want to measure in an unknown samples. So I choose a principal component analysis, which is the basic algorithm used in unsupervised methods, and I will show you a few examples how you can use it to get more out of your data. So first example is data from mice model of depression, which is being developed by Ewa Bączyńska at the Nensky Institute of Experimental Biology in the group of Professor Jakub Włodarczyk. So when you develop a new depression model, you have to check if it works well. So for that, you can weigh the mouse before the experiment and after the stress protocol. You can do for swimming test and check how long it is just floating. You can check if it still prefers the sucrose, sweetened water, instead of normal one. And you can check if it still prefers to stay in the dark or it just doesn't care anymore. Apart of that, you can do experiments on microscopic level, so you can just visualize your neurons and count dendritic spines. You can do biochemical analysis of blood or mass spectrometry. And which one of those you should choose to say if your mouse is anhedonic or resilient? So it's very hard to choose the best model, the best measurement. And it's not only about the choice of the measurements that you will make, but also which value you should choose. So if you check literature, some people check how many times the mice changed its mind during the experiment. So how many times it started to swim after it was floating, or how many times it changed from the dark to the light again. Or other people check the time. So what was the total time the mouse was floating? Or what was the total time the mouse was in the light compartment? And you can do that in the first 2 minutes, in the first 2 minutes after it started to do something, after the latency period, and there is no actual recipe for that. The other problem, if you do so many measurements, how you choose your cutoff value? So for one measurement, it's easy. We can always do that. There are methods for that. But if we want to do all of those measurements, what is the best way? And here we can use principal component analysis. So if you look on two-dimensional data, you can see that the highest variance of the data is in this direction, U1, and the second orthogonal direction, which you have the highest variance, is this U2. So basically what principal component does is just shifts your axis to the direction of the highest variance. It may not seem as something important when you have two-dimensional data, but if you have a lot of measurements and a lot of different variables, then you can combine different variables in just one new direction that will describe best your data. From principal component analysis, you get a lot of different information, but two plots are especially important. This is the loadings plot, and this is the plot of how important is your variable for your analysis. So if your variable is a near 0, 0 point in your new axis, it doesn't give you much information. So you can probably discard it, and nothing will change. And here are different data obtained from those 4 experiments. And from 4 swimming tests, you have, here are the data that are just measuring the total number of time the mice was floating. And here are the total number of times it changed its mind. And also the different time intervals from 0 to 2 minutes, from 2 to 4, and so on. So you can see that it's good to check both the number of transitions and the total time of floating, because they give different information, and it's best to get this information from all experiments, not only from a small interval. It's also interesting that the latency from dart-like box and the latency from for swimming tests gives the same information. So regardless which one you get the same amount of variance. And choosing those parameters, those new variables, we were able to do this. So this is an unsupervised method, so we didn't specify which samples are from which group. We just color-coded them. And the algorithm was able to perfectly distinguish from control animals, resilient, that suffered the stress protocol but didn't undergo depression, and the one that were anhedonic. And now the next step is to join results from different experiments and try to use this model to predict unknown samples. The next example is based on data measured by Magdalena Kundis, who is here in the room. And Magdalena is working on a new electrochemical setup which is able to measure very small amount of liquid from 40 to 70 microliters in a system that is called rotating droplet. So we have a rotating rod with 2 electrodes and a working electrode on the bottom, and we rotate a small droplet of liquid between them and measure the signal. The objective of this study is to measure dopamine and serotonin simultaneously in presence of interfering agents such as uric acid and ascorbic acid, which are often found in body fluids. And there are some mechanistic questions. For example, how fast we should spin the droplets. It is not quite sure because, we get higher current, but we also get higher noise if we increase the velocity. And there are also some other problems. If we have only serotonin and ascorbic acid, uric acid, there is no shift of peak. But or with dopamine, there is also no shift for uric acid. But if we have serotonin, dopamine, and both acids, there is clear shift of the peak of uric acid. So it's very hard to think of quantifying unknown mixtures that have all those 4 compounds. And here are results of principal component analysis again. We were able to distinguish samples that had a mixture of dopamine and serotonin and only dopamine, and samples that didn't have neurotransmitter were clearly left outside of clusters. And maybe it's not so remarkable if you don't look at this graph, but here you have uric acid and ascorbic acid, so you have a clear peak for uric acid and a small one for ascorbic acid. And here is the same sample, but with 10 nanomolar of dopamine and serotonin. And I don't know if you can, but I cannot see the dopamine here, or neither the serotonin here, but the algorithm can. So you can clearly go down with your sensitivity. So again, a next step is to use another algorithm to use this model to quantify those samples, but for that we have to measure different concentrations of uric acid to account for this shift in peak position. And if you don't do this kind of studies, why you should know something about this kind of algorithms? So a few years back, there was a study of a dead salmon that was put in an fMRI, and people were showing different pictures and asking what's your emotional state. And in some cases, the salmon actually had an emotional state, the dead salmon. So if you have 100,000 of samples, then even if a very stringent confidence interval, you will be able to show some statistical significance. And that's why it's always, I'm sure you are sure about your data, but it's always better to check twice what you are reading in the literature. And thank you for your attention.
Thank you very much, Emilia. You will, well, the group will make our life easy, I hope, so with all the data analysis and with these machine techniques that we could apply. Thank you very much. Do we have questions to Emilia? If not, then I would like to thank you again, and I would like to thank all the speakers in this session and also you, the audience, for being here with us, and I hope you enjoyed this session and you found these lectures interesting. Thank you very much.